florets are undergoing anthesis in this rhythmic daily cycle. And they're coordinating growth so that florets of a different age are anthesing at the same time. And so let's see. If we take a step back, before anthesis, the sunflower capitulum really shows just this continuous age gradient of florets, with the oldest florets on the outside and the youngest florets on the inside. And what's very apparent are these spirals of florets that are also called parastiches. And those arose due to the spiral phyllotaxy initiation pattern on the shoot apical meristem. But during anthesis, that pattern converts to discrete rings of florets opening. And these rings I call pseudo whorls because they're not a real whorl, because a real whorl would imply that these florets are of the same age, but they're actually of different ages, but they're somehow coordinating growth to happen at exactly the same time. And I want to just highlight that this is pretty unique because most inflorescences that show spiral phyllotaxy, such as our favorite model plant, Arabidopsis, what you see is that the oldest flower undergoes anthesis at an earlier time than the next oldest flower, and then the next, and the next, and the next, and the next. It's not often that you see coordination of florets opening at the same time, even though they're a different age. So if we just take a moment to really define the different stages of anthesis in sunflowers. Pre-anthesis, the floret is all encapsulated in this corolla tube. And if you dissect out that corolla tube, what you see is the anther tube subtended by the filaments. And collectively, we call this the stamen tube. And within that anther stamen tube is the style. So then in the next stage, the staminate stage, what happens is that the filaments grow rapidly and push the anther tube up and over the corolla tube. And what you're not seeing is that simultaneously, the style is also growing up alongside the filaments at this time, but still hidden within the anther tube. Then in the pistillate stage, you have the style continuing growth, and it's in this way that actually the pollen is pushed out of the stamen tube and presented. And that's where pollinators come in and take all the pollen. And then the style pushes up and unfolds the stigma, and that's where pollinators can also then spread the pollen to the different stigmas, promoting outcrossing. So finally, in our post-anthesis state, we have the ovary with the fully anthesed floret. Oh, sorry, I did want to just point this out. What you see in this pistillate stage is that actually the filaments shrink quite a bit compared to in the staminate stage. So when we are asking about anther elongation in earlier talks, this is kind of why I was interested on the timing of it. All right, so if we go back to this video and look at sunflower anthesis in photoperiodic conditions, you see that the first stage is this kind of swelling of the florets, followed immediately by the staminate stage and then the pistillate stage. And this proceeds throughout the life of the capitulums until all the florets are used up. And we, as a lab, decided to further measure this. And what my first observation was is in that initial step where you see the florets kind of swelling, it's actually not because of corolla growth or any other growth of the top part of the floret. It's because the ovaries elongate very rapidly. And that pushes the floret up above the surface of the capitulum. And I don't know this for certain, but I'd like to test it. That might have some, give access to pollinators to access like the nectary or the pollen. And what's really interesting is that you see this coordination of growth from one pseudowhorl to the next, so that florets in a post-anthesis or during anthesis stage, they have much longer ovaries, and then you have this sharp step down in size for the pre-anthesis florets, which have shorter ovaries. So in the Harmer lab, we took many capitulums and grew them in different environmental conditions and growth chambers, and took time-lapse videos, and we scored 3 stages of anthesis. The first being that ovary growth. The second being the stamen emergence up above the corolla tube, which is actually due to filament elongation. And then finally, the style emergence. And what we found is that in photoperiodic conditions, so cycling light-dark conditions, every 24 hours, you have a new pseudowhorl of florets undergoing anthesis. And it's in a very coordinated, tight timing. Now, I was interested to look at what's happening within that pseudowhorl of florets. So what I did is in these time-lapse videos, I followed a continuous parastachy of florets, with the oldest florets on the outside and the youngest florets on the inside. And again, this represents an age gradient of florets. And I scored them for ovary, stamen, and style growth versus time. And what you see is that florets within a pseudowhorl exactly coordinate their growth. So this exact coordination of growth, even though they're a different age, kind of hints at us that something is regulating it. It's not a sand timer of age. And of course, our favorite regulator of time is the circadian clock. So what is the circadian clock? Inputs from the environment, such as temperature cues or light, dark cues, are transmitted to this central oscillator that exists in plants, but also, pretty much every single biological organism out there. And this central oscillator is a complex network of genes and proteins that feed back on each other and control downstream processes. And these downstream processes we call outputs. And those can be things such as, hormone signaling, response to stress, metabolism, or flowering patterns. And it's, very beneficial to have a circadian clock because you're having a fitness advantage so that these processes are not on all the time. And they're also matching the environment to happen at the exact time of day that's most beneficial. But as a circadian biologist, we ask, how do you determine if a biological process is regulated by the circadian clock, or if it's merely being driven by environmental cues and, a direct response to the environment? So what we do is we test certain conditions and tests. And the first thing is we ask, does this biological process persist in constant conditions? So we ask that with anthesis rhythms. And to do this, we first transferred the capitulums from photoperiodic conditions to constant light conditions and constant temperature. And what we found is after 1 day of normal anthesis, then the pattern of anthesis is completely disrupted. So the first most obvious thing is that the stamen emergence is completely abolished, so that you have absolutely no pollen presentation or stamen emergence. And you also see that the ovary and styles grow in this continuous manner. And on the capitulum, what that looks like is this age gradient where you no longer see discrete pseudowhorls of florets opening. You just see them opening consecutively. And while I haven't measured this, I just want to say that observationally, much less pollen is presented in this condition, likely due to the kinetics of the stamen tube not being grown up and out along with the style, pushing the pollen out. Now, another proxy of pseudowhorl abolishment is if you look at the ovary growth. So a cross section of this capitulum looks something like this, where you see this continuous transition of the ovary length. And that's in contrast to in the photoperiodic conditions where you had that step down in ovary growth from one pseudowhorl to the next. And, that other test I did where I looked at individual florets along continuous parasticles and scored them for anthesis timing, you see this diagonal pattern that really matches the age of the floret, in contrast to in the light dark conditions where you saw that, horizontal time, or I guess it's vertical time, sorry, of timing being exact. So from this, we can say, wow, that's really interesting. Constant light and temperature disrupts normal anthesis patterns, but it's not very informative of whether the clock is regulating anthesis patterns. So we next looked at anthesis rhythms in constant dark conditions. And luckily for us, what we found was that anthesis not only proceeds normally, but it proceeds in these coordinated pseudowhirls happening or undergoing anthesis every 24-ish hours. One thing to note is that style length or growth is really slowed down in constant dark, so that the styles of one pseudowhirl, instead of completing growth within 24 hours, goes on to the next day. The style start time, though, is relatively coordinated the first 2 days, but then gets out of phase. So really, we're seeing also differential regulation of floral organs, hinting at maybe the clock is regulating this process, but there's other processes involved. Now, another indication that pseudowhirl coordination is preserved in constant dark is by looking at the ovary length. And what you see is that the ovaries from one pseudowhirl to the next show this step down in size, very similar to the light-dark conditions. And I'm sorry if there's some yabbering in the background, but my son just woke up. So anyways, yeah, also the ovary length is much less diminished, or it's very diminished in constant dark. And that shows that, there might be less sugar right away. All right, so great, that's a tick on the clock might be regulating anthesis patterns. And the next property we measure is temperature compensation. So something great about the circadian clock is it temperature compensates. And that means that when you transfer an organism to different temperatures, as a good timekeeper, a circadian clock would maintain rhythms exactly the same time, which we call periodicity. So the time it takes to go from one cycle to the next. And something that's temperature compensated will show the same period over a range of different physiological temperatures. And that's in contrast to, say, an enzymatic process, which will be much slower in the cold and warmer in the, or faster in the warm. And what we see is that in 3 different temperature conditions, anthesis of ovary and stamen growth, and this was in constant dark, showed temperature compensation. So great, we have 2 things that really are strong criteria for a circadian clock regulated process. And the final thing I want to show you is the concept of gating. So the circadian clock has this ability to gate certain biological processes so that the level of response to environmental cue is time of day dependent. And that might mean that if you give a cue, such as a dark pulse or a heat pulse in the day, you might get a different response than if you've given it during the night. And to test this, we took photoperiodic and trained sunflowers, and after the first day of anthesis, transferred them to these different conditions where they received a dark cue during what the plant thinks is a subjective night versus a dark cue where the plant thinks it's a subjective day. And we looked at anthesis patterns. And what we found was that when the sunflower received a dark cue during its subjective night, the phase of ovary and stamen, I'm not showing you style, but it's very similar to stamen timing. It doesn't change much. There's a little bit of phase change, but it's pretty normal. But when you give that dark cue during the subjective day, what you see is that the pseudowhirl delays anthesis. So specifically, if you look at the ovaries in these 4 different condition, you see that it starts ovary growth. And then it just kind of extends it and pauses it and delays it until it gets that second dark cue that kind of is like, oh, okay, this is when we're going to open. And then it proceeds through normal anthesis. But the takeaway from this is there's a time of day sensitivity response that is dependent on whether it's a subjective day or night cue. And again, this is a great indication of circadian clock regulation. All right, so in conclusion, the first thing we did was kind of observe anthesis patterns in sunflowers. And what we found is that the sunflower development transitions from this continuous age gradient of florets before anthesis with, oldest florets here and youngest here, to during anthesis, you have this discrete rings of florets opening every 24 hours. And that's true in photoperiodic conditions and constant dark conditions. But there's something about the constant light that masks or, disrupts normal anthesis. And secondly, we've determined, and there's many other tests that I'm not showing you, that the circadian clock regulates sunflower anthesis patterns, but that's most likely in conjunction with floret age and communication between florets in a pseudowhirl. And what I mean by this is that, these florets in, say, this pseudowhirl 1, which are in a post-anthesis state, might send some sort of signal to these florets in the next pseudowhirl, and in conjunction with the appropriate age and signals from the circadian clock allow transition to anthesis. But simultaneously, those are sending some repressive signal to the younger florets along with age, saying, wait, don't transition to anthesis until we've completed anthesis. So what's next? This is the genetic section of the conference, but I haven't talked to you much about specific genes. So we're trying to determine what is the genetic basis of floral organ elongation in sunflower florets, and what is the genetic basis of the sunflower circadian clock. And we're doing that with very painful and meticulous time course dissections of floral organs that we are sequencing. And also we're asking in field trials, is the timing of sunflower anthesis as regulated by the circadian clock important for attracting pollinator visits to the field? And, in simple terms, this just means if we have a sunflower whose circadian clock is out of phase with that of the clock of a pollinator, and we put them all in the field, is a pollinator visits going to be disrupted? So with that, I want to say thank you. And because it's the time of COVID and we don't have lab pictures, I am showing you pictures of my lab mates that I can take as many pictures of as I want. And thank you to my lab, Stacy Harmer, Veronica Thompson, who is the grad student who's helped me a lot with RNA-seq, along with all my lab members, and my collaborators, such as Nikki Crew and Ben Blackman, and of course, the funding sources and my university. So with that, I'll say thank you.